Muscular contraction is a detailed process. First of all, a nerve impulse must be sent through central and peripheral nervous systems until it reaches a neuromuscular junction. At this point, a complex process sees the impulse go through the junction and reach the muscle fibers it needs to affect. This is where the sliding filament theory takes place. First of all, a stimulus, in this case a football, must be presented. This stimulus is detected by sense organs such as the eyes. This information is passed through the central nervous system to the brain via sensory neurons. The brain processes this information and sends an instruction in the form of an electrical impulse down the spine, which is part of the central nervous system, and out through a motor neuron, which is part of the peripheral nervous system. The electrical impulse arrives at a neuromuscular junction in the form of action potential before it can be passed on to an effector muscle. This is what a motor neuron looks like. Dendrites on the nerve cell collect signals from the rest of the body. These impulses are passed to muscle fibers along an axon. A fatty myelin sheath speeds up this process, allowing the impulse to jump down the axon. When the impulse gets to the fiber, it must pass through a neuromuscular junction, shown on the diagram. When the impulse gets to this neuromuscular junction, it is picked up by synaptic vesicles at the bottom of the motor neuron. The impulse then diffuses across the gap known as the synapse in chemical form, and is then picked up on the other side. The impulse is sent down transverse tubules, which are deep tunnels running into the sarcolemma. This is where the sliding filament theory comes into play. The stripy appearance of muscle fibers results from the regular arrangement of the myofilaments with myofibrils. In the myofibril arrangement, the sarcomere is one of the segments into which a fibril is divided. This is shown in the image. The M-line provides an attachment for myosin filaments. The Z-disc provides an attachment for actin filaments. The A-band is the darker band of the myofibril containing the myosin filaments. The H-zone is the lighter section in the middle of the A-band where only myosin is present. The I-band is the lighter band of the myofibril containing only the actin filament. When the muscle is relaxed, the protein tropomyosin blocks the actin binding site, or ABS. This means that the myosin heads cannot bind with the actin. Muscle contraction occurs when the muscle receives an impulse from a nerve cell in the form of an action potential. The impulse penetrates to the center of the muscle fiber via T tubules, deep tunnels, into the sarcolemma. This triggers calcium ion channels in the sarcoplasmic reticulum membranes to open. Calcium ions diffuse out rapidly into the myofibrils to bind to the troponin molecules. The troponin molecules change shape, causing the tropomyosin to move to a different position on the actin filaments. The actin binding site is now free, and the extended myosin heads can bind to the actin. At the end of a contraction, the bound ADP and phosphate are released, causing the myosin heads to change back to their original shape. This pulls the actin filaments towards the center of the sarcomere. The binding of another ATP molecule to each myosin head causes them to release the actin. The breakdown of this ATP to ADP plus P in the myosin head releases energy which allows the head to extend once again. During muscle contraction, the actin and myosin filaments do not shorten, but instead they slide past each other. If calcium is still present and there is still a supply of ATP, then this process is repeatable again and again. In a relaxed muscle, after the action potential is passed, the calcium ions are pumped back into the sarcoplasmic reticulum. This allows the troponin to return to its previous state and causes the muscle to relax. There are three main types of muscle contraction. These are concentric isotonic, eccentric isotonic, and isometric. First of all, concentric isotonic. Isotonic concentric contractions involve the contracting muscle shortening. An example of this is the upwards motion of a bicep curl, in which the biceps brachii shortens as it contracts. The next example is eccentric isotonic. Isotonic eccentric eccentric contraction involves the muscle lengthening. An example of this is the downwards movement of a bicep curl as the biceps brachii lengthens. The last main example is isometric contractions. Isometric contractions involve no movement in the contracting muscle. Holding a weight in place when performing a bicep curl means that the biceps brachii is isometrically contracted. There is no movement. One last type of contraction is isokinetic contraction. This causes the muscle to shorten as it contracts, but it requires a constant speed over the entire range of motion. An example of this is an arm stroke when swimming. The even resistance of the water offers a constant speed at maximum contractions. This was Sliding Filament Theory and Contractions. Thank you for listening.